Hello students, welcome to grade 12 biology. Today we are going to see how sex is determined, sex determination. But before we go directly to this lesson, which is lesson 2, let's revise what we have seen in the previous lesson. In the previous lesson, we have seen genetic crosses. We have seen monohybrid cross and dihybrid cross. We have seen that monohybrid cross is a type of cross, genetic cross, that involves only one trait, only one character, either seed color or stem size and this. Whereas the hybrid cross is a cross that involves two traits simultaneously at the same time. For example, seed color and seed shape could be taken at the same time, and this kind of genetic cross is known as the hybrid cross. Today, we are going to see how sex is determined. Now let's start our lesson by an activity. Students, try to answer what is sex determination. What do you understand by sex determination then? How do you understand? Well done. Sex determination is a system in biological system that determines development of sexual characteristics in an organism. And this occurs during fertilization, union of gametes, fertilization. So sexual characteristics in organism is known as sex determination. Now let's see very important terminologies which are very useful in our subtopic. Karyotype is a photograph of all chromosomes in a cell arranged in a homologous pair. You know that chromosomes are arranged in a homologous pair, and the photograph of all the chromosomes in a cell arranged in a homologous pair are known as karyotype. Sarai gene is the dominant gene on the Y chromosome. You know that there are two types of sex chromosome, X chromosome and Y chromosome, and this is dominant on Y chromosome that causes a mammal to develop as a male. The other is sex-linked genes, which are found on the chromosomes that determine sex. Let's see chromosomes and genes related to sex determination. Sex is determined by a specific chromosome, and these sex determining chromosomes are X chromosome and Y chromosome. For example, in humans, let's take humans as an example. X chromosome and Y chromosome are the two chromosomes that determine sex. Now let's take male and female, mother and father and mother. Males have X and Y chromosome, whereas females have X and X chromosome. So during fertilization, the possibility is 50% of having an offspring child of male and female. So we can have XX, which is a girl, or we can have XY, which is a XY, which is a boy. So sex is determined in humans in such a way. Now, human karyotype is a complete set of chromosomes in a cell of an organism, organized and displayed in standard format. Karyotype shows a visual representation of the number, size, and shape of chromosome. The number of chromosome, size, and shape of chromosome is represented by karyotype, enabling the analysis of chromosomal abnormality and sex determination. So karyotype is very important in determining sex as well as analysis of chromosomal abnormalities, either in number, size, or shape. The key features of a karyotype are chromosome composition in humans have 46 chromosomes. 22 pairs of these are autosome, so they are body cells or non-sex chromosome. One pair is sex chromosome. This sex chromosome is XX for females and XY for males. So these are known as autosome. So that body, whereas one pair, we have seen and we have said that the chromosome number of chromosome in humans is 46. 23 comes from the mother and 23 comes from the father. So these 22 of them is autosome, non-sex chromosome, and one pair is sex chromosome. This is XX for females and XY for males. Karyotyping is used for identifying presence of X and Y chromosome. It also differentiates between male and female karyotypes. Sex determining gene are genes that are important and expressed in a Y chromosome. They are located on the Y chromosome. Their key role is determination in sex determination by initiating development of male characteristics in mammals. It is located on Y chromosome, specifically in the short arm YP. It's absent in females because females have XX chromosome, no Y chromosome in females. The testes produce testosterone, which masculinizes the body. So they initiate this hormone. Anti-Mullerian hormone, AMH hormone, is also secreted, preventing the development of female reproductive structures. So this is very important gene. Absence of this gene leads to the development of ovaries and female phenotype, which is default pathway. And mutations or change in this gene can result in disorders such as XY gonadal dysgenesis, Sawyer syndrome. Individuals with XY chromosome develop a female characteristics. A person who have a male chromosome, XY chromosome, develop a female characteristic due to defective gene, SRI gene. Also, X-sex reversal, an XX individual, SRI translocation may develop male characteristics. The opposite may also occur. A female may develop a male characteristics, or a male develop a female characteristics, which is very difficult and difficult syndrome. The role of this gene is on the Y chromosome. It triggers or initiates the development of testes by initiating and triggering the development of testosterone, which is a very important hormone, and development of gonads are also activated by this gene, testis formation, and this gene is absent in ovaries or in females. XY chromosome, X carries many genes vital for survival. Y is smaller and contains fewer genes. When we compare the two sex chromosomes, X and Y chromosome, X chromosome carries many genes compared to that of Y chromosome, whereas Y chromosome contains smaller in size as well as fewer in number, fewer genes. Now let's proceed and see sex chromosomal abnormalities. What are the abnormalities of this? One is non-disjunction during meiosis can lead to abnormalities like Turner and Klinefelter syndrome. These two are because of non-disjunction during meiosis. Let's see what is Turner syndrome. Monosomy or X0, sterility may occur, shorter stature, and some masculinized features may occur during this syndrome. Klinefelter syndrome is trisomy, whereas in the previous Turner we have seen monosomy, that is one, but here we do have 3 chromosomes, XXY chromosome. These also may lead to sterility and also feminized features in males. Males may develop a female characteristics due to this trisomy, this syndrome. Other systems of sex determination. In birds, we do have ZW for females and ZZ for males. Temperature dependent sex determination in reptiles are dependent on that of temperature. Now it's your turn to answer. What are sex influenced and sex limited traits? Sex influenced traits and sex limited traits. Well done. Sex influenced traits are traits that are more likely to occur in one sex but may sometimes occur in the other sex. So there is a possibility to occur in both sexes, and they are known as sex influenced traits. Most likely they occur in one sex, but sometimes it's possible to occur also in the other sex. Pattern of boldness is another example of often this boldness is a phenomena that occurs in male. It's an example of sex influenced trait that occurs in males. Sex limited trait is a limited trait that occurs only in one sex. Limited means it's limited into one sex, and never occurs in the other sex. Sex limited trait occurs only in one sex, never in the other sex. Mind you, always you have to remember male sex and female sex. Sex limited traits occur only either in male or in female, never in both. Sex limited are only expressed in one sex. Both males and females have genes that stimulate the lactation, but these only expressed in females. the condition of cryptorchidism and descendant testicles is genetically determined, but clearly can only ever be expressed in males. It's a kind of trait that's expressed in one sex, never in the other. This brings us to the end of today's lesson. Again, goodbye until we meet in the next lesson.